Hey guys, Mrs. Horn here. Today I'm going to talk about 4 organic molecules. By the end of the notes, I'm hoping that you have learned the characteristics of all 4 molecules. You should be more familiar with what the structure looks like, what elements make up those molecules, functions of the molecules, and different groupings of the molecules. We will need a textbook for today's lesson, so if you want to pause the video, grab the textbook so that it's ready, that may be a good idea. So let's get started. Before we talk about the large organic molecules, we need to discuss 2 processes, one of which is a building process and the other is a breaking process. The term dehydration synthesis refers to the synthesizing or building of a large molecule from small pieces, from small monomers. Monomer is a really small piece of a large molecule. And you make those large molecules, those big polymers or polypeptides or polysaccharides, you make them by putting small pieces together. Dehydration synthesis, in order to do that, you have to remove water. So you take out a hydrogen, you take out an OH, and by doing that, it frees up some bonds on those molecules, and they're kind of empty and waiting to attach to something else. So those molecules would actually attach to each other and therefore make a larger molecule. Now, the opposite of this would be hydrolysis. We could also call hydrolysis digestion. Hydrolysis, if you break the word apart, hydro means water, lysis means to break apart. If we can remember that, that should be our trigger to remembering that hydrolysis is the breaking down of a large molecule into small molecules or monomers. And in this process, water is added in order to do that. Other things about this, for both of these processes, enzymes are required. These things cannot happen without the help of enzymes. The first organic molecule that we're going to talk about are carbohydrates. There are 3 different levels of carbohydrates, and when I say levels, I mean 3 different sizes of carbohydrates. The first is called a monosaccharide. This is a monomer. Glucose is an example. The purpose of glucose, it's used in cellular respiration. It is the chemical energy that is used to fuel the process of cellular respiration. This is a process that happens in the mitochondria. We haven't talked in detail about that yet, but I know you've heard the term cellular respiration. And the purpose of that respiration for a living cell is to make ATP. ATP is energy. It stands for adenosine triphosphate. That energy is used by the cell to carry on its life functions. So it uses it to move things across the cell membrane. It uses it to move things through the endoplasmic reticulum. It uses it to synthesize molecules or break down molecules. So the energy that it makes is really, really important, and it makes it because of that glucose, which is a monosaccharide. There are other monosaccharides, but glucose is by far the most famous. As we get to a bigger carbohydrate, we're going to call this group the disaccharide group. Now, carbs typically will be in the shape of a ring. We talked, we will be talking later in the notes, we talked about it in chapter 4, that's where it was, the carbon skeletons, and we said that they could be linear, they could be branched, and one of the options was they could be in a ring, and carbohydrates typically are shown in a ring. A monosaccharide has 1 ring, and a disaccharide has 2 rings. The example that I have here is maltose. Maltose is made up of 2 rings. It's made up of actually 2 glucose molecules. Maltose we can find in your food, so you would obtain this from your diet. And then that maltose, being too big to do anything, your body has to break it down. So it breaks it down through the process of hydrolysis. Maltose is made through the process of dehydration synthesis. But your body is going to then take that maltose and break it down because glucose is what your body can use pretty easily. I would not say maltose is the most famous of disaccharides, but I chose this example because it's made up of two glucose molecules, and that's easy to remember. Probably a more famous disaccharide would actually be sucrose. Sucrose is table sugar. I had a few examples here of polysaccharides. Now, polysaccharides are made up of many rings, so all these green rings here. So we're talking 3 or more rings for polysaccharide. Starch is a pretty well-known polysaccharide, and your cells are going to use that starch and store it as energy. Later, cells might pull from that energy reserve and break it down into glucose and then use that for cellular respiration. Cellulose is another example of a polysaccharide. Cellulose, we're going to find a lot of that in plants because cellulose makes up the cell walls of plant cells. Your cells don't have a cell wall, so that's why we're not going to find it in your cells. Cellulose is the most abundant organic compound on the planet, which does make sense if we consider what we learned in ecology, that our producers and autotrophs are the most abundant. They have the greatest biomass, they are the greatest in number, they have the greatest amount of energy because there's a ton of them. So it makes sense that there's more cellulose than any other organic compound. Glycogen is another one. Glycogen you would find in animal cells. Your body will store it in your liver, and it's going to stay there until your body needs it. So if your pancreas detects that you have low blood sugar, it will send a message to the liver telling your liver to, hey, take some of that glycogen and break it down into glucose and then put that into the blood, and then we're going to be okay. So that's what glycogen does. Let's move over to lipids. Lipids are made up of glycerol and fatty acids. Those are the smallest components of lipids. Those would be the monomers of lipids. You can't get any smaller than that. However, once you see a picture of what they look like, glycerol and fatty acids together, looks really huge. It's way bigger than a carbohydrate monomer or a protein monomer. Lipids are also hydrophobic. I do not want to mix with water. Good examples are oils, butters. I have a picture here of the difference between a saturated fat and an unsaturated fat. So let's stick over here to the left. At room temperature, a saturated fat is at a solid. Butter is a great example. It's solid because the molecules are very closely packed together. And if we look closely at the structural, the way that the structure is put together, it would kind of all look in a straight line. Everything's a single bond. Whereas an unsaturated fat at room temperature is more of a liquid. It's a liquid because the molecules are more loosely packed, so there's more space in between them. The reason being because we have some double bonds in here. So that double bond makes pretty significant difference. Looks very different than this formula over here. And rather than being butter, it's oil. But either one, whether it's saturated or unsaturated, both are hydrophobic. Lipids in your body. A group called phospholipids. I've got 2 groups that I want to talk about here. Phospholipids is the first one. Phospholipids make up the cell membranes of all cells. All cells in your body, all cells in plants, bacteria, fungus, all cells of all living things are made up of phospholipids. And phospholipids are made up of a hydrophilic head and then a hydrophobic tail. And if you look here, there's actually 2 layers of them. So I have the heads on the top with the tails, and then kind of the underneath of the tails and then the heads. This whole section in the middle is hydrophobic, and that's why it's kind of hiding in the middle. It doesn't want anything to do with water, so it hides in the middle. And the hydrophilic heads are on the outside, so those are exposed to water outside the cell, and these heads are exposed to water inside the cell. This is a great example of how form determines the function. So the way that these are organized, the way that they have a shape, determines what they can and can't do. Another type of a lipid that I want to be familiar with is a steroid. There are actually naturally occurring steroids in your body. Cholesterol is an example. Cholesterol is also found in the cell membrane of animal cells, and it helps to do two main things. It helps to keep the cell consistently fluid, not really fluid, not flimsy and liquidy, but allows the cell membrane to move a little bit, have a little flexibility, a little elasticity. It also helps the cell to be semi-permeable, so we'll let some things in, some things out, but it's not a free-for-all. Things cannot just come and go, whatever it is, whenever it wants. So cholesterol is a very important lipid on the outside of the cell membrane, helping to control these characteristics. Well, let's move over to proteins. Proteins are our third type of organic molecule. It's made up of amino acids. Amino acids are the smallest form of a protein, so amino acid would be the monomer of a protein. When you put a bunch of amino acids together, you get a polypeptide. Proteins are also enzymes. I don't have that written here, but an enzyme is a protein. So when we look to classify an enzyme as an organic molecule, it would be here. Here's where you're going to need that textbook. And the next section here, so these 4 boxes on this page, and then there are 4 boxes on the next page, we're trying to figure out what do proteins do? What are the functions of proteins? So these 4 boxes, these 8 boxes altogether, you're going to find those on page 78 in the textbook. And I want you to label what each is and give me a description of what it's doing. So if there's one here that's an enzymatic protein, tell me what an enzymatic protein does. So now would be a good time to pause the video and take 8 to 10 minutes and fill in the name of those proteins and tell me what it does. Okay, so you've got the names of all 8 of those proteins. We know what they do, and now we're going to move on, and we need to know that there are more than just 8 types of proteins. There's and all those proteins are made up of different amino acids, right? Those are the building blocks of proteins. There are 20 amino acids. They're not all naturally occurring in your body. Some of them you're going to have to get from your diet. And I've listed 10 here. A lot of them end in an "ene," like a valine, leucine, phenylalanine, tyrosine. A lot of them end sounding the same. Tryptophan is one here that doesn't really sound like anything else. Usually you hear of tryptophan around Thanksgiving time when you're eating turkey, because there's lots of tryptophan in your turkey. Remember those pipe cleaners we got out last week? And we took the pipe cleaners and we had a straight pipe cleaner that represented the primary structure. Then the secondary structure, we took that same pipe cleaner and we coiled it around our pencil. And then for a tertiary, we took the pipe cleaner and then we bent it so that it was no longer a straight curlicue, it was more of a bent curlicue. And then to get a quaternary structure, remember what we did? We took that pipe cleaner and then you combined it with somebody else. And then we took all the pipe cleaners at your whole table and put them together in a big mosh pit of pipe cleaners, and that represented the quaternary structure. Well, for this section of the notes, you're going to be in your textbook on page 82 and 83, and I want you to go through and identify the different structures of proteins, which I kind of just gave them to you, and you're going to pull out from those pages the structures, sorry, not the structures, but the function or some characteristic of each of those proteins. So on this page and over to the next page, there are 4 different structures of proteins. Write the name of the protein and give me the function or some characteristics of what that protein does. Take about 5 minutes and get through that chart, and then we'll finish up. Okay, we are in the home stretch here. We've talked about carbs, we've talked about lipids, we've talked about proteins, and now we're on our 4th organic molecule, nucleic acids. Nucleic acids are made up of nucleotides. So a nucleotide is a monomer. There are two types of nucleic acids. There's RNA, and then the more famous of the two would be DNA. DNA we can find in the nucleus of every cell of a living thing, and DNA is what makes RNA. DNA, as it makes a copy of itself, its copy is called RNA. And RNA, which it's made in the nucleus, but it's a small enough piece where it can leave the nucleus and then it goes out into the cytoplasm somewhere and helps to make proteins. And those proteins, which remember are built from amino acids, a lot of those proteins are enzymes or any of those 8 different types of proteins that you just put in the chart. It makes those proteins, and then those proteins now have a very specific function and purpose in the cell. I mentioned that nucleic acids are made up of nucleotides, but what's a nucleotide made up of? Nucleotide is made up of 3 things. So although a nucleotide is the smallest component of a nucleic acid, a nucleotide is made up of a nitrogenous base, a sugar, and a phosphate. I have some pictures of those at the bottom. But this nucleotide is the smallest form of both DNA and RNA. A nucleotide is a monomer of a nucleic acid, just like amino acid is to a protein and a monosaccharide is to a carbohydrate. So that is our information on organic molecules, their characteristics, their structures, and their functions, and I hope this helps.